But there's some really interesting hints of therapeutic effects of CBG in the literature, two of which are quite important and intertwined. The first is its potential analgesic effect. And as we talked about before, our lab is very interested in the analgesic effects of cannabinoids and also its potential appetite stimulating effects. And the reason why this is important is because currently there aren't very many therapeutics that are approved to improve appetite. And the FDA approved medications that are on the market aren't so great. And in fact, THC is one of these FDA approved medications. But as we know, THC does have those adverse effects that can limit its clinical utility.
Welcome to High Five with Dr. Igor Grant, Distinguished Professor of Psychiatry and Director of the Center for Medicinal Cannabis Research at UC San Diego. Each episode will take you on a fascinating journey into the evolving world of medicinal cannabis, as Dr. Grant poses 5 thought-provoking questions to his guest, uncovering their personal journey and unique contributions to the field. Now, here's your host, Dr. Igor Grant.
Today, I'm especially happy to welcome my colleague, Dr. Ziva Cooper from our sister university at UCLA. Dr. Cooper is the director of the UCLA Center for Cannabis and Cannabinoids in the Jane and Terry Semel Institute for Neuroscience and Human Behavior and professor in the Department of Psychiatry and Biobehavioral Sciences and Anesthesiology at the David Geffen School of Medicine. Her current research involves understanding variables that influence both the therapeutic potential and adverse effects of cannabis and its constituents through a translational approach that includes double-blind, placebo-controlled studies. Welcome, Ziva.
It's so great to be here, Dr. Grant. Thank you for having me.
Hey, if I call you Ziva, you got to call me Igor. All right. So as you know, I'm going to ask you a few questions so we can talk in a little bit of detail about the work you've been doing. So your research has focused on both the possibilities and drawbacks of using cannabis and its constituents, for example, the minor cannabinoids and terpenes as medicines. You've also looked at possible differences in effects between men and women. I'd like to ask you a few questions about each of these areas. So, one of your areas has been examining how men and women might respond differently to cannabis, both as a medicine and as a recreational drug. Can you tell us something about your insights in this area?
Yeah, so this area stemmed both from the recent trends in seeing the uptick of medicinal cannabis use among women, alongside really interesting preclinical, so animal laboratory data demonstrating the biological underpinnings for differences between male and female laboratory animals in response to a tetrahydrocannabinol specifically. And so this became of interest because of survey after survey demonstrating that although in the past, when you looked at the prevalence of cannabis use among people in the United States, usually males outpace females 2-1. But when the survey started coming out with respect to who's using medicinal cannabis, it was clear that women were using medicinal cannabis as frequently or even more frequently than males. And some questions came to mind. So, there's obvious questions that maybe this is a societal or a cultural shift that drew women, females into using cannabis as medicine. But when you dig into the preclinical literature, you find that there are profound differences between male and female animals in response to tetrahydrocannabinol that might hint towards improved therapeutic outcomes among females for biological reasons. Those biological reasons being perhaps circulating hormone fluctuations across menstrual cycle phases. Also, how males and females are breaking down THC in very different ways that can have potentially clinically significant effects with respect to pain relief specifically and other outcomes. So this is kind of why I got drawn into the area. Igor, my training was in preclinical psychopharmacology. So that was, when I was doing my PhD, I really focused on animal models of drug action, behavioral pharmacology. And so I've always been steered in certain questions or certain research questions in the clinical lab based off of a lot of what we find in the animal literature. So it was this really exciting area where we were able to translate or think about how we can translate from the preclinical literature and answer some really important questions from a public health perspective.
So maybe I could just follow up. So we know, for example, that there are different sensitivities that men and women have to the effects of drugs like alcohol. Okay. Is it the case here that women respond to a different dosage level of THC, if we're talking about THC specifically, or that certain syndromes are more likely to be responsive or not responsive in men and women? Can you talk about that for a minute?
Yes. So, in our own research, we found one of the hypotheses was that women would be more sensitive to the pain relieving effects of cannabis with THC in it than males. And that was in part based off of the animal literature.
Mm-hmm.
What we actually found when we first started doing this work was surprisingly, when we compared women and men in our studies, the women actually showed very little analgesic effects when receiving smoked cannabis compared to males, which was very different than what had been seen in the animal literature. And we kind of dug into our data and we realized that, well, the people who we were looking at were people who were using cannabis every day. So they were what's called, frequent or heavy, using cannabis heavily. And looking back at the literature, we saw that, in animals, although females are more sensitive to the pain relieving effects of THC at first exposure, they develop tolerance to that effect very quickly, as opposed to males who don't. They do develop tolerance, but just not as quickly. And so we felt like that we were kind of seeing that in our people in our studies because they were daily cannabis users. And so that led us to do an interesting study where we were looking at differences between males and females volunteers and looking at differences in people who were using cannabis once a week, so kind of occasional use versus people who are using it every day. And to see if we can demonstrate that maybe females who don't use it that often are more sensitive to the pain relieving effects of cannabis than males. But with the heavier users, we would start seeing that tolerance. And, we're just digging into those data now. We have a pretty large group of people that we've run through the study, and we are seeing that females seem to develop tolerance to the pain relieving effects, and the females, when they're just using occasionally, they may in fact be more sensitive to the pain relieving effects than males. And what's interesting about that is that we're controlling for parts of the menstrual cycle phase when certain hormones are at higher levels, and we think that those hormones might contribute to the outcomes. Probing that will be a different study. So this is pain, and pain is one of the primary reasons why people use medicinal cannabis. And so with respect to understanding cannabis therapeutic effects, we've chosen to kind of focus on pain. But, there are many other indications in the epidemiological literature and the survey literature, and now starting to emerge in the clinical trial literature, demonstrating differences between males and females. And not just with respect to therapeutic outcomes, but also potential adverse effects. And so, Igor, I think that from your group, Michelle Sexton, if I recall correctly, I think it was Michelle Sexton and some other groups from Johns Hopkins, they did a study where they were looking at withdrawal symptoms between males and females. And so we know that people who use cannabis regularly, cannabis with THC in it, there's a subset of people that do experience withdrawal when they stop using it. And what these studies have found was that there were different profiles of withdrawal symptoms that occur between males and females. And so in that respect, it's important to understand what the potential differences are so that, people who are going through withdrawal or who are trying to achieve some state of abstinence might be aware of what they could expect. Some Other interesting literature that has come out over the years, again, is differences just in the acute effects of cannabis with THC. And so something that has popped up is that females might be more sensitive to the anxiety promoting effects of certain doses of THC compared to males. And again, this is important if you think about it from a public health perspective. If females are starting to use cannabis or cannabis derived products to help with certain indications, understanding that it's very important to start at low doses and that the anxiety promoting effects of cannabis might be greater among females than males.
Yeah, so that's really interesting. So, if I can kind of encapsulate it, sounds like from the animal literature, you would have expected greater sensitivity in women. Is, well, it depends on how experienced they are with cannabis in the first place, as an example. And then, as you pointed out, some of the untoward effects may be different as well in women. So, okay, very good. I'd like to migrate to another topic if we can, unless you want to say something more about this.
No, that's great. Thank you.
Okay, so much of the research, of course, on cannabinoids as medicines has focused on molecules like THC, tetrahydrocannabinol, which is the main psychoactive ingredient in cannabis, and also cannabidiol, which, although not psychoactive in the usual sense of the word, has been, thought to be of help maybe actually in managing anxiety or epileptic disorders and things of that nature. But of course, there are many other constituents in cannabis, including other cannabinoids or THC-like molecules, and also other molecules such as terpenes. And I know some of the work you've been doing and has been done at your center has now been looking at these minor cannabinoids and terpenes. Can you say a word about that?
Yes. So in general, cannabis is a very exciting plant because, as you said, there are two chemicals in the cannabis plant that we know we're getting to know a lot about. If we don't already know a lot about them, there's delta-9 tetrahydrocannabinol, THC, and cannabidiol, CBD. But in addition to those, there's hundreds of other chemicals. And some of those chemicals have been probed a little bit, maybe in chemical assays or in preclinical models, and some of them show promise for potential therapeutic effects. And some of those molecules, unlike delta-9 THC, it's suggested that those molecules might provide some type of therapeutic benefit while not having the same type of psychoactivity or intoxication as delta-9 THC. And so, I think that CBD, people first became very excited about CBD. Well, it's been studied since the 1970s, but around 2008, people became much more interested in cannabidiol. There had already been quite a bit of preclinical studies on cannabidiol, though that has grown exponentially. And I think part of this excitement about cannabidiol was the fact that it is from the cannabis plant, but it's not intoxicating. It does not have, abuse liability like delta-9 THC. It does not have cognitive impairing effects. It really didn't seem to have any of the adverse effects that we know about with delta-9 THC. And so it kind of became like a safe cannabinoid to try or to potentially test for therapeutic endpoints, some of which were already kind of being probed in the preclinical literature. So CBD isn't alone in the cannabis plant for these types of features. There are a couple of phytocannabinoids in the plant, minor cannabinoids in the plant that we are particularly interested in. We're very lucky to have received support from the CMCR to study one of these phytocannabinoids called CBG, Cannabigerol. And Cannabigerol is actually the phytocannabinoid that is kind of like the mother of all cannabinoids. And this is where CBD and THC come from as the plant matures and the phytocannabinoids are synthesized. And what's interesting about CBG is that, again, unlike delta-9 THC in animal models, it is not thought to have those psychoactive, intoxicating effects. It's not evident that if you take large doses of CBG or if an organism is exposed to large doses of CBG for long periods of time, that there will be a withdrawal syndrome that occurs. But there's some really interesting hints of therapeutic effects of CBG in the literature, two of which are quite important and intertwined. The first is its potential analgesic effect. And as we talked about before, our lab is very interested in the analgesic effects of cannabinoids and also its potential appetite stimulating effects. And the reason why this is important is because currently there aren't very many therapeutics that are approved to improve appetite. And the FDA approved medications that are on the market aren't so great. And in fact, THC is one of these FDA approved medications. But as we know, THC does have those adverse effects that can limit its clinical utility. And one thing that's important about improving appetite or thinking about a medication that improves appetite, especially in a population that has chronic pain, what we see in populations with chronic pain is usually appetite suppression or reduced appetite is usually associated with severity of chronic pain. And so the two kind of go hand in hand. And so if you have a therapy like THC, for example, that can both improve appetite and reduce pain, you're really hitting those two issues head on and potentially improving quality of life, symptom management, so on and so forth. Now, we already talked about the issues with THC. CBG in animal models, there's some hint that it can help to improve appetite while also reducing pain, but unlike THC, not have those adverse effects. And so we have a study where we're looking at CBG. When we proposed the study, I think it was the first study under placebo-controlled conditions looking at CBG's acute effects. So we were kind of testing out the waters with respect to what type of dose might have an effect. What effects might we actually see in people? Would we see the same types of things that we see in animals? What's the time course? What mode of administration might we see these effects? And so we base things off of, the doses that we see in the animal literature and also just based off of like what people are generally exposed to when they use cannabis. And we did that specifically for safety to start with pretty low doses. And the study is designed so that we can look at both CBG by itself and look to see how it affects appetite, how it affects eating behavior, and also how it might affect pain response. And this is done in healthy participants. Again, it's one of the first times that we're looking at CBG in people. And so we first want to look at CBG in healthy people before we move it to a patient population if we have a signal in healthy people. And so we're able to look at appetite stimulation in healthy people. We just feed them a very small breakfast and then we withhold food for a period of time and we're able to see that appetite or hunger at least. And then we're able to give participants an option to choose whatever type of food they want to for the rest of the session. And we can look at the types of food, the caloric intake, the macronutrient content of the food that they're taking for the rest of the session based off of the study drug condition. And so we're looking at CBG by itself and we're looking at THC by itself to compare the two. And then also we look at the combinations of CBG and THC. And the reason why we look at the combination is because there's an idea that CBG might be able to enhance the pro-therapeutic effects of low doses of THC without having the adverse effects that are typically related to THC. So it's a fairly large study that the CMCR is helping to fund, or not helping, they are funding. And we're able to look at several different endpoints across several different doses. We're collecting blood so that we can look at potential pharmacokinetic interactions between CBG and THC that might help to inform behavioral outcomes that we might see.
So like you point out, there are people who really need to have their appetite stimulated because of either chronic disease or some other situation. And whereas THC does do that, it does produce a high in most people, and one may not want that all the time. And so Cannabigerol may be one of these things. It would be interesting to see whether if you give choices to people, whether the THC people gravitate to Cheetos or Doritos versus the Cannabigerol people going to more healthy foods.
Yes, I mean, yes, it would be interesting to see that. And Igor will be able to answer that with this because what we do is we withhold food for a period of time and then we give them a list of all the food that we have. And Igor, we have the Doritos, we have the ice cream bars, but we also have the almonds and the apples and the peanut butter. And so we'll be able to look at just general food choice, but also, the caloric intake and we do measure the amount of the food that is eaten after it's ordered. So that's CBG and it's really exciting. We're almost done with that study. So it'll be nice to look at all the data together. There's another interesting cannabinoid that also has some good preclinical evidence supporting its potential pain relieving effects without its THC-like intoxicating or abuse-related effects. And that's CBC, Cannabichromine. And so this is a phytocannabinoid that we're really interested in. It's been studied actually in a number of animal studies, also back from the 70s. There's an interesting signal that CBC might also be helpful for pain, and it's thought to act on a number of different pharmacological targets. So not necessarily like the CB1 receptor like THC, but it's thought that it might alter endocannabinoid tone by changing the way endocannabinoids are metabolized and synthesized. And it also might be acting at pro-inflammatory sites to help reduce inflammation and some other really interesting sites. And so CBC preclinically shows some really nice promise, but similar to CBG, there have been very few studies in humans. We're hoping to bridge that gap as well to be able to test and look at if there is a signal for the pain relieving effects of CBC in people, and if it could potentially increase very low doses of THC's analgesic effects and reduce those adverse effects. So that's another phytocannabinoid that we're interested in.
For our listeners, when you say phytocannabinoids, you mean a plant-derived cannabinoid, right?
Yeah. And so frequently we use cannabinoid as shorthand, but there's all types of cannabinoids. There are synthetic cannabinoids. There are synthetic cannabinoids that are not in the plant. There's synthetic cannabinoids that do exist in the plant but are made in the lab. And there are semi-synthetic cannabinoids that are made from plant material. But these are actual chemical compounds that are found in the plant. So those are phytocannabinoids.
Now, we also have terpenes, which are actually probably the molecules that give the particular aroma, shall we say, to different kinds of marijuana. Isn't there some work also suggesting that the terpenes might also have beneficial effects?
Yes, so terpenes, there's many different terpenes in the cannabis plant, and there are about 9 that are present in relatively high concentration relative to the other terpenes there. And so some of these terpenes also, you look at preclinical studies and there's really interesting data suggesting that they might be helpful for pain. So we're looking at 2 in particular. One is called Myrcene and the other one is called Beta-Caryophyllene. And actually CMCR is helping to support some of the preclinical, some of the animal studies that are looking at some of these terpenes here at UCLA. In humans, we're doing studies now to look at these 2 terpenes in very similar models to what we're looking at with CBG, where we're trying to understand the terpenes by themselves, whether or not they are analgesic, can they reduce pain without that intoxication and abuse liability that we get without the 9-THC, and also can they augment the effects of THC to boost the pain relieving effects and reduce the adverse effects. And thus far, we're showing some interesting signals with beta-caryophyllene. They're pretty low doses, actually. And we use a inhaled model. So people are inhaling these terpenes. And we're seeing hints that beta-caryophyllene might, in fact, produce analgesic effects. We're not seeing necessarily the same type of effect with myrcene, but both of these terpenes do seem to not have those psychoactive effects that THC elicits. So that's good because we know that people are starting to use these terpenes. They're available in dispensaries and in smoke shops. Different companies are kind of leveraging the terpenes when they're making their formulations. But it doesn't seem thus far like, at least at the doses that we are looking at, which are relatively low, that we're getting adverse effects. And in fact, with beta-caryophyllene, we might be seeing reductions in pain with administration of beta-caryophyllene. And, Igor, it's interesting. Just over the last few years, we've started to see a increased interest of studying these terpenes in the preclinical studies. And I think in part, they're nice compounds to study because you don't, as a preclinical researcher, you don't have to deal with all the regulatory hassles that come along with studying, THC, or you need a Schedule 1 license and you have to find a source that also has, that is federally compliant. Terpenes, like you said, terpenes are in many different fruits and vegetables and herbs and spices. And so preclinical researchers are able to study these terpenes without the regulatory red tape. And so that has led to, I think, some really nice pharmacology that has emerged in the preclinical space, sophisticated pharmacology to understand how these terpenes are working, what type of pain might they be helpful for, what other effects might they have? There a potential anti-anxiety like effect or antidepressant like effect? So I think over time, we're just starting to see a surge of this type of research. And over time, we're going to have really nice data to turn to that can inform clinical work.
Can I just maybe ask a different question? Actually, it's related, but you sort of commented a little bit on how these minor cannabinoids might have, if you will, a THC sparing effect. That is, reduce the amount of THC you need or reduce the high or something. But there's been a lot of work also on the cannabinoids as opioid sparing, right? And what we usually mean by that is people with more severe chronic pain, find opioids like oxycodone certainly to be effective, but they're also quite addictive and can lead to all sorts of problems. And there's been some suggestion that THC and other cannabinoids might reduce the need for these opioids. What can you say about that? And also whether these minor cannabinoids or terpenes might have similar properties?
Yeah, so this is a really exciting area of research. And in fact, Igor, I think that CMCR funded that 2007 paper with Donald Abrams, kind of was like the first study, right, to look at combinations of cannabis with THC and an opioid in a pain patient population. Donald was able to demonstrate that there was increased analgesia when adding the THC. I think that was one of the first studies that manipulated both cannabinoid exposure with opioids to look at outcomes. But if you look at the animal literature again, there is really nice behavioral pharmacology demonstrating this synergy between specifically THC and opioids, suggesting that there is this type of opioid sparing effect of THC. And other work later on has been looking at cannabidiol. So Sarah Jane Ward at Temple University has been looking at the potential for cannabidiol to do this as well. And we had a study in 2018 that examined the potential opioid sparing effects of smoked cannabis with THC and found that there was potential synergy with combinations of cannabis with THC and a very low dose of an opioid that doesn't usually produce pain relief. When we combined the two, we saw pain relief above and beyond what you would see with THC by itself or what you would see with that low dose of opioid by itself. And that led us to different studies looking at combinations of THC and CBD to understand the opioid sparing effects, and other studies also looking at potential opioid sparing effects of terpenes, which in the preclinical literature also demonstrates that some of these terpenes might have opioid sparing effects. So since then, though, there's been some really interesting data. The group from Johns Hopkins published a study recently looking at cannabidiol and opioids, demonstrating that there might be beneficial effects of combining cannabidiol with opioids. But at this point, the literature is still kind of mixed. And I think that we're still trying to figure out, under rigorous double-blind placebo-controlled conditions, the extent to which cannabinoids, THC specifically, other cannabinoids might have opioid-sparing effects and how that could be applied in the clinic. But if you look at survey data and if you look at kind of registry data, there are indications that people who have chronic pain, who are using opioids and choose to use cannabis, there is a subpopulation of that group who are able to either reduce their opioid use over time or even cut it out, totally. Now, there's also survey data and epidemiological data that shows that, this effect isn't universal, and that's not always the case. And it could be that, some people who are using both might actually increase their use of both over time, which is not good, right? That's an adverse effect. And so I think it will take time to parse out what are the variables that might contribute to this beneficial interaction over time. And so this is, Igor, you bring up this like opioid sparing effect of cannabinoids, but I think, as a field, we're also becoming interested in other ways that cannabinoids might help to reduce other types of drugs that also have adverse effects. So for example, benzodiazepines, right? There's this interest that cannabinoids might also be helpful in reducing benzodiazepine use for a variety of reasons.
Things like Valium, right?
Yes, Valium, like for anxiety, and things like that. So, there's also a body of literature, interestingly, showing that even though cannabis is classically a drug of abuse, that cannabis might actually be a helpful therapeutic for substance use disorders, right? So we know that there's studies looking at cannabidiol in people with opioid use disorder. There have been studies with THC and CBD in combination, or THC by itself, in people with cannabis use disorder, and the potential for these cannabinoids to have a potentially therapeutic effect in people with substance use disorders.
There's actually some interesting preclinical data to suggest that cannabinoids, both CBD and possibly THC, have these anti-craving aspects. And it's like with weight loss, almost anybody can stop using drugs for a period of time, but then there is this craving that develops that makes it hard to maintain sobriety. And there's some evidence that cannabidiol particularly might be helpful in that. Some data actually with nicotine models in animals. So yeah, so that's really interesting. Just talking about the pain though, and I had this interesting exchange with Mark Ware, whom you know, who's a Canadian pain doctor, among other things. And we talked about, well, there's this kind of desire to undo the psychoactive effects of THCs, kind of separate that out from the pain effects. But he kept pointing out, pain is a psychological experience as much as anything. And so it may be that in some cases you don't want to get rid of that so-called psychoactive effect. And some people have reported that if they use things like THC, they still experience the pain, but they don't care about it so much, or it kind of is tolerable. I don't know if you have any thoughts about that.
Yeah, this is an interesting point, the degree to which the positive feeling, we always ask people, when we expose them to THC, cannabinoids, we ask them to rate their mood and how much they like the drug or what kind of an effect they are feeling. Is it a good effect? Is it a bad effect, right? So is it really bad if there's a good effect associated with the THC administration, if it's helping with the pain? Is it really bad if they are somewhat intoxicated? At what point is it bad? And at what point could it be clinically helpful? Right. And so I think another way to look at this is that, pain itself is impairing at a certain point. It's impairing. THC is impairing as well. There's been some interesting work demonstrating that people who have chronic pain who are taking THC are actually less impaired. Their quality of life might be better. So is there a balance here? We're looking at healthy people primarily. We do have studies that focus on pain populations and other type of patient populations, but we're looking at healthy people. We might not be, when we're looking at intoxication and impairment, understanding how that translates to the pain population in a very clinically meaningful manner will be absolutely critical, down the road in determining how cannabis and cannabinoids can be used as medicine. And for some populations, the intoxication or the impairment that people have from THC, if it is reducing those people's pain, like for example, in palliative care settings, is it a bad thing? Is intoxication or feeling good a bad thing? But if you're talking about somebody who has chronic pain and who's going to work and needs to perform at a certain level, and the intoxication is getting in the way of their performance and their daily function in a professional environment, well, then that is a different type of context and a different question.
So it's very interesting, and it depends on the context, of course. As you say, in a palliative care setting, it may be very different. Or even in not such a serious situation, if a person doesn't need to drive or operate complicated machinery or something like this, maybe that's okay. Well, our time is moving on, and I'm wondering if I could go to a more of a kind of wrap up question, which is, if you were queen and could just order whatever you wanted to be done, are there thoughts you have about how to advance and clarify, the place of cannabinoids and terpenes in medicine?
And so I really appreciate this question, and I will answer it from a place of doing this research day in and day out, and dealing with the regulatory hurdles and challenges that I encounter from just accessing the drug to be able to answer what I think we all think is an important question for health, for public health, for protecting the public, for potentially developing new therapeutics, to the everyday regulatory work that we have to do as clinical researchers. Now, I say that knowing that, the regulations are very important in protecting patients and participants who are in our studies, but I do wish that one day there could be more efficient pathways or more study drug available to researchers, because the time and effort and cost entailed with doing this particular research, I think, is above and beyond what is typical for clinical research in general. Clinical research is hard, right? Whatever you're looking at, whatever patient population you're looking at, whatever study drug you're looking at, it's going to be difficult. There's a whole nother level when you're studying cannabis and cannabinoids. And so if we can establish more efficient pathways to getting this work done, that would be great. And I'm saying this, Igor, as somebody who's been doing this for a pretty long time. So anybody who wants to get into it, they have to go through the learning curve and get the licenses, get the approvals, and that itself can take an enormous amount of time, right? So it's really a hard field to be into. It is extremely gratifying, and also it's important work. It's also very fun, like talk about the people that you've had on this podcast, the people who you're hoping to have on, and they're awesome. I mean, how lucky is it that we get to interface with these scientists who are asking important questions and figuring out creative ways to be able to answer those questions. So it's wonderful. It's a blessing, but those regulations could definitely be a curse at times.
Yeah, and just to also clarify further, the regulations, many people say, well, the Schedule 1, when you call cannabis the same thing as heroin, basically, that's a problem and it's difficult. And that's true. But that's not the only thing. And you alluded to the fact that you're studying, for example, minor cannabinoids, but you have to get Food and Drug Administration, FDA approval, and they require that these things be manufactured to certain standards and such. So that's another whole issue, isn't it?
Yep. Yeah. It's not just the Schedule 1 issue. It is getting drug that is compliant with the FDA standards. And again, it's important. We definitely don't want to give study drug that's contaminated to participants, especially if they're patients. But, access to that drug is really hard. When CBD became no longer scheduled, we thought, oh, it's going to be so much easier to study it, right? But how many sources do we have for CBD that meets FDA requirements and is also affordable, right? So that's also an important point, is that it costs a lot of money to produce these study medications that meet FDA standards and that are also affordable for researchers.
Well, good wrap up. It's wonderful to talk to you, Ziva, and thank you so much for taking the time to participate in this podcast. Is there anything else you want to say by way of wrap up?
For the moment. This has been wonderful. Thank you so much for inviting me to speak on your podcast. And again, we are very grateful for CMCR support and that you're producing these podcasts. Hopefully it will help educate people and maybe it will help to turn things a little bit. All the queens and kings that you have on, I'm sure they're sharing their ideas of what their wishes are, and maybe one of those wishes will come true.
Very good. Thank you so much for being with us.
Thank you.
Yeah.
If you like this podcast, please subscribe so you don't miss future episodes. For more information on the Center for Medicinal Cannabis Research at UC San Diego, please find us on social media at CMCR_UCSD on Twitter or X, Instagram, and other social media platforms. Or visit our website at cmcr.ucsd.edu. Thanks for listening.